Most welcome to this press conference. I'm Göran Hansson, Secretary of the Nobel Committee. The Nobel Assembly at Karolinska Institutet today decided to award the Nobel Prize in Physiology or Medicine 2009 jointly to Elizabeth Blackburn, Carol Grader, and Jack Szostak for the discovery of how chromosomes are protected by telomeres and the enzyme telomerase. And I will now give the word to Professor Rune Tofsgaard of the Nobel Committee, who will provide an overview of the laureates and their discoveries.
As you just heard, this year's Nobel Prize is awarded for the discovery on how chromosomes are protected by telomeres and the enzyme telomerase. Now, what is a telomere? Major scientific progress during the first half of the last century have established that the genetic material, our genes, are encoded by DNA. The chromosomes in the cell nucleus here, they contain the DNA, and the telomeres are the ends of chromosomes. They are here depicted in yellow. It was also noted that the telomeres had a special nature and seemed to be important for chromosomal integrity. It is an amazing fact that a complete set of genetic instructions on how to build a multicellular organism is contained in every cell. Thus, when a cell divides and the genetic material and the chromosomes are copied, they must be completely copied to preserve the genetic material and avoid breakdown. If the chromosomes would not be completely copied, as depicted here on the left, the chromosomes and the chromosome ends will get progressively shorter with successive cell divisions, and in the end, there will be loss of genetic material and severe damage. However, in most cases, this is not the case, and what one sees is rather a maintenance of the chromosomes. With the knowledge available in the late 1970s, it was a big mystery how the chromosomes could be copied to the very end. When a cell divides, both strands of the DNA thread, here depicted in blue, needs to be copied. And with the metaphor, copying proceeds like on a highway with uninterrupted traffic in one direction, whereas in the other direction, the copying proceeds in a stepwise fashion. And starting platforms, also called primers, are made as needed. Subsequently, these primers on platforms are removed, the gaps are filled in by DNA, and the different pieces are stitched together. However, at the end, when this starting platform is removed, the gap cannot be filled. And upon successive cell divisions, the chromosome should get shorter and shorter. And this is the end replication problem. How this could be solved puzzled scientists for a long time, and many different solutions were proposed. In the late 70s, Elizabeth Blackburn studied Tetrahymena thermophila. It's a single-cell organism living in water. And she found that the chromosomes and the chromosome ends especially had a specific structure. They contained a repeated DNA sequence like this. Jack Szostak, working at another university and addressing a different topic, made artificial mini-chromosomes that he introduced into yeast cells. The problem was that these artificial mini-chromosomes were rapidly degraded and could not be maintained. The breakthrough occurred after Elizabeth Blackburn and Jack Szostak met at a conference in 1980. And they then decided to test whether or not telomere DNA from Tetrahymena could protect chromosomes in yeast. So to do this, they took these mini chromosomes and they put telomere DNA from tetrahymena on each end of the chromosome. And the result were striking. When these were introduced into yeast cells, now the chromosomes could be maintained in a stable fashion for a long time. And this was in direct contrast to what happened in the previous experiments with mini chromosomes without telomere ends, where there was rapid degradation. So with that, they had found that telomere DNA from one organism, tetrahymena, protected chromosomes in an entirely different one, yeast. And this demonstrated a previously unrecognized fundamental mechanism. Given the importance of telomeres, the next obvious question was, how are telomeres formed? Is there an unknown enzyme? Carol Grader and Elizabeth Blackburn started a search for the enzyme. They wanted to see if they took a cell extract from a tetrahymena organism, mixed that with synthetic DNA sequences corresponding to the telomere DNA from tetrahymena, followed by addition of building blocks for DNA, nucleotides. Could then telomere DNA be extended? Some of these building blocks here were also radioactively labeled to allow later visualization. Carol Grader got a very valuable Christmas gift when she, on the Christmas day 1984, went to the lab to analyze results of assays. And she saw results like this. So this ladder-like banding pattern represents different lengths of telomere DNA where repeat units have been added. And the difference in size between each band here corresponds to the extra addition of one block of telomere DNA. So they had then discovered that there was an enzyme activity. They also found that the new enzyme contained a protein component, but how did it work? It should be noted that it is the upper parent strand of the DNA, the telomere DNA, that is actually extended. And how could now the order of the building blocks that should be added be determined so that you get a specific sequence? In continued work, they found that the new enzyme contained a necessary RNA component. When they could isolate, clone, and sequence the RNA component, it turned out that it had a sequence motif like this, which could recognize, pair, and position the enzyme just at the tip of the telomere DNA. And subsequently then, this RNA component serves as a guide, the template. So now the order of the building blocks that are being added are determined by the sequence here, the RNA template. When one set of building blocks have been added, the enzyme here, the protein, moves to the new end of the telomere DNA, and then another block of DNA sequences are added. And this way, the process continues. With that, the telomerase was discovered. And telomerase is a unique enzyme that extends telomere DNA. It prevents chromosomal shortening and instability. A fundamental mechanism like this has broad medical implications. And one may say that primarily in three areas, there are important considerations for cancer, certain inherited diseases, and for aging. Cancer cells divide frequently, and they need to preserve their telomeres. And it was quickly discovered that cancer cells have abnormal telomeres, and that most cancer cells have a high telomerase activity. So this raised hopes that new therapeutic strategies to treat cancer could be developed by targeting telomerase-expressing cells. And such therapies are presently under development, and clinical trials have been initiated, but no conclusive results are available as of yet. As expected from a fundamental mechanism, if there is a defect, this would cause disease. And in fact, mutations in both the protein and RNA components of the telomerase have been found in a group of related hereditary inherited diseases. The disease manifestations are primarily seen in the bone marrow, the lung, and the skin, and they are associated with a disturbed maintenance of stem cells, and in most cases also associated with short telomeres. In particular, in bone marrow, low telomerase activity leads to anemia. When it comes to aging, it was also in the early days found that telomere shortening is correlated to a reduction of lifespan in cultured cells. And conversely, that increased telomerase activity extends cellular lifespan. So it was then speculated that telomere length and telomerase activity may be the main determining factor in aging. However, we now know that in the organism, aging is really a complex process that is influenced by many different factors. Telomere length and telomerase activity is one of many factors that are affecting aging. In summary, Elizabeth Blackburn, Carol Grader, and Jack Szostak discovered how telomeres protect the ends of chromosomes, identified a new enzyme telomerase that synthesizes telomere DNA, and they thereby solved a long-standing fundamental problem in biology. These are the laureates. Elizabeth Blackburn, now working at University of California, San Francisco. Carol Grader, working at Johns Hopkins University School of Medicine in Baltimore. And Jack Szostak, working at Harvard Medical School, Boston. More information can be found at the homepage of the Nobel Foundation.
Thank you very much. The panel with members of the Nobel Assembly are now ready to take questions. And let me just first introduce the panel. We have Rune Toftgård, whom we just heard, expert in molecular biology and cancer research. We have Nils-Göran Larsson, pediatrician with expertise in genetics and mechanisms of aging. Urban Lendahl, expert in genetics and developmental biology. And we, of course, have the secretary of the Nobel Committee and Assembly, Göran Hansson, expert in cardiovascular research. My name is Claes Kärre. I'm the chairperson of the Nobel Committee this year. My field is immunology. I'm afraid that we have a problem with the microphones, so you will have to shout the questions. And I should say that we are prepared to take questions in Swedish and English, and if you go slowly, also in German and French, but we will answer all questions in English. Please. The question was it difficult to arrive to this conclusion? Every decision for a Nobel Prize is a long journey to carefully assess the weight and importance of the discovery and the contribution of the scientists who made the discovery. Yes? Why did it take so long time, 25 years? Perhaps one should say that the first discovery in this series described by Rune Toftgård was made 30 years ago, and the last of the critical experiments was made 20 years ago. So why has it taken so long time? The answer for that is that it's a maturation process of a scientific discovery. It's not so that something happened this year or last year that finally made us decide, okay, now this is ready. But it has been a slow maturation process, like for many other Nobel Prizes. And let me elaborate on that a little bit. There are two phases in this maturation process. One is that the scientific community has to recognize the discovery, the generality of the discovery, and start to nominate the scientists. Because without nominations, the Nobel Committee cannot work. Secondly, the Nobel Committee has to evaluate the discovery. Is it so much more important than other discoveries? And who are the persons who made the key discoveries? And that's the evaluation phase. So that, on average, takes 20 years. Last year's prize to the viruses had matured for 20 to 30 years, and the knockout prize the year before had also matured for that time. I think sometimes the medicine committee is said to be slower than the other committees, and I think that there is a reason for that. Biological and medical discoveries usually take longer to mature because the findings have to be generalized in many other systems, and biological experiments and medical observations usually take longer time. But there are examples of Nobel Prizes in medicine and physiology where the process has been much more rapid, 10 years, even 5 years.
Do these combined discoveries mean that the process of aging of human beings will be controlled so that the average age will amount to 150 years for men and 170 for women?
So the question is, can we use this discovery to control aging? I think our expert in aging, Nils-Göran Larsson, can comment on that.
Yes, well, I think we should remember that aging is due to multiple types of damage to our bodies, and then there are different repair and maintenance systems, and telomerase and telomeres is just one of these maintenance systems. But certainly during human aging there is a shortening of the length of telomeres, and there is also some data to suggest that renewal of tissues by stem cells may be dependent on telomeres and telomerase. So I would say that the current data certainly suggests that telomeres and telomerase are implicated and of importance, but it will not be the only explanation to the aging process.
Where were the people who got the prize when you reached them this morning or before getting here, and what did they say? How did they react?
Göran Hansson.
They were at home, all of them, and I was lucky to reach all of them. They were a bit sleepy when they got the call, but they were very happy and look forward to come to Stockholm in December.
More questions? Yes? I think it's the first time that the prize in physiology or medicine is awarded to 2 women at the same time. One more question?
What is the next step within this area of research?
I think there are many steps. Why don't we first comment on cancer research?
I think in terms of cancer, the clinical trials will be very important, and it will also be very important to assess potential side effects, since also telomerase activity is found in stem cells. One must be careful in fiddling around with the activity of telomerase in this way. Also, more recently, it has been found that some components, or in particular the protein component of telomerase, also has effects that are not linked to the telomerase activity as such. So there might also be unexpected new findings coming up.
Göran Hansson?
Yes. There's a lot of excitement now about a potential role for telomeres and telomerase in a broad variety of diseases, and there are statistical associations between telomere length and stress, heart disease, diabetes, and so on. Now it will be very important to figure out what is real and what is mechanism here and what is, so to say, statistical noise. So that will be important to clarify from a medical point of view. And again, as Rune Toftgård said, the clinical trials that are ongoing will teach us probably a lot about how treatment directed against telomerase will affect the human body.
Nils-Göran Larsson, do you want to comment also?
Well, I think the discovery of telomeres and telomerase is a very fundamental mechanism in the cell, and such fundamental mechanisms will very likely lead to profound medical importance in the long run.
Is there yet any quick test for telomerase
gimmick. Maybe a commercial?
I mean, one has to be really careful because the inter-individual variability is quite big. So you cannot use it in an individual to assess the rate of aging in a particular individual. It only sort of works in larger materials, right? So I wouldn't recommend you having your telomeres tested.
If there are no more questions, I think it's time to close this session of the press conference. We thank you very much.